All right, hello everybody. It is Sister Culbertson here, and I want to go over how to do lesson one practice problem number 4. Okay, so this is the one that is asking you about sister chromatids and the number of chromosomes at different phases of meiosis and the cell cycle. Okay, so before we jump into it, we need to understand a few things about what we're talking about here and about chromosomes and what constitutes different types of chromosomes. So the first thing you need to know about, this helps me visualize it a little bit better, is a karyotype. So a karyotype is the number and visual appearance of chromosomes in one cell. Okay, so here we have the number of chromosomes. You see there's 22 pairs plus an X and a Y that we also call a pair. So 23 pairs, which would mean that there are 46 chromosomes. So this is representing the human genome. Um, okay, so let's take a look a little bit closer about what we're talking about. So you see that these are all numbered 1 through 22. So those are numbered based on homology. So we call these homologous chromosome pairs. So you can see here the total number of chromosomes. We have 23 pairs of homologous chromosomes. So in each one of these pairs, one chromosome is maternal, meaning it came from your mom, and one chromosome is paternal, meaning that it came from your dad. So if we look up here at chromosomes pair number 1, you can see that these chromosomes are very similar. One of them is maternal, one of them is paternal. Okay, so what does that mean, a homologous chromosome pair? So this means that each of these pairs are similar in size, banding pattern. So you see that in chromosome 1, they look like they're pretty similar in length, right? So these are a lot longer than, say, chromosome set number 3. Okay, but even within these, there's a little bit of difference in their size. So it looks like one of them might be a little bit longer. Their banding patterns might be a little bit different. So there's a big bright spot on this one, and there's not a corresponding bright one on this one. But you can see that they're more similar than maybe if you compared one of these to a chromosome in set number 2, where there's a lot more green banding present and a lot less of the really bright red banding. Okay, so that means that it's similar. They have the same genes on them, but the makeup of the genes is a little bit different. Okay, so there's variation within the genes that are on each of these homologous chromosomes, but they contain the same set of genes. All right, so that's a homologous chromosome pair, and they are similar, whereas sister chromatids are identical DNA molecules. And these are linked together at the structure called the centromere. So these definitions are from problem number 1 in that same assignment. So that's where you can find these. Okay, so how do we get identical DNA molecules? So if they're identical, they can only form after the DNA was replicated, or in the S phase of the cell cycle. So each homologous chromosome in the pair, so one being maternal and one being paternal, will replicate itself to form these two sister chromatid molecules. So if you look over here at chromosome pair number 4, what will happen is the maternal one, so we'll call it this one on the left side, will replicate itself, and it will stay attached to that replicated chromosome through a structure at the center called the centromere. Okay, so this arm is identical to this arm. They're not similar, they're identical. Okay, and the same will happen with this paternal one over here. Okay, so it will replicate itself and stay connected to that replicate via the centromere. Okay, so you can see now that these two are identical to each other, these two are identical to each other, and these are still similar in nature. Okay, so in this representation right here, each one of these arms is considered a sister chromatid. Okay, so this maternal copy of the homologous chromosome pair has two sister chromatids, 1, 2. And then this paternal copy in this homologous chromosome pair also has two sister chromatids, 1, 2. So in total there are 4 sister chromatids, but there are only 2 homologous chromosomes. So these are still connected to each other, so these two sister chromatids together are considered one molecule still. And the same is true for these two. Okay, I think this is where most people get confused, so it's really important that you understand this concept of sister chromatids and how they replicate. These don't just smash together and form another one that has similar chromosomes in it. They're actually identical. So rewatch this slide if you need to. Okay, so let's get into question 4, part number 1. So this is asking you about a chimpanzee, and they tell you that it's a diploid animal that has 48 chromosomes in it. And they're asking you about the different total number of chromosomes, pairs, and sister chromatids prior to DNA replication, okay? So I've just made a little representation of what a karyotype of the chimpanzee would look like. So we have the 23 sets of chromosome pairs and then an X and Y. And it's going to be the same thing that we talked about earlier. So for the total number of chromosomes before DNA replication, that's the same as what they've already given you. So that's going to be the 48. Okay, and then moving on to the second part of the question, how many homologous chromosome pairs are there? So in order to figure this out, you need to know if the chimpanzee is diploid or triploid or haploid. So we know from the question that it's a diploid animal, meaning that there are 2 sets of each chromosome, okay? So chromosomes 1 through 23, there are 2 copies of each of those. That's what that means. So the total number of chromosomes is 48, and you'll divide that by 2, because we know it's a diploid, to get the number of pairs. So that'll be 24. Okay, so if this happened to be triploid, we would divide it by 3. But it's diploid, so there you go. Okay, so then it asks about the number of sister chromatids. Well, we said sister chromatids are identical, right? So they have to be replicated. And this question is saying that they're asking about what's going on prior to DNA replication. So there's not going to be any identical chromosomes in the cell. They're only going to be homologous pairs. Okay, so the answer to that would be 0. Okay, moving on to part number 2. So in this question, they're asking about the composition of the cells at the beginning of metaphase 1. So in order to answer this, you need to know what's happening at metaphase 1 and where that lies within the cell cycle. So metaphase 1 is after DNA replication has occurred, but before sister chromatids have separated into separate cells and before homologous chromosome pairs have separated into cells. Oops. So for the total number of chromosomes, each of these have undergone replication, right? So you have one molecule that has the 2 sister chromatids, but they're still attached to each other and still considered one molecule, okay? So you're still going to have a total of 48 chromosomes or 48 molecules within the cell. Okay, so for the number of homologous chromosome pairs, like we said, the centromeres are still attaching those two sister chromatids, so they're still considered one molecules. So you'll still have the same number of pairs, which is 48 divided by 2, which is 24. And then finally, how many sister chromatids are present after DNA replication has occurred? So each of these 48 molecules that we have over here has replicated itself and has 2 sister chromatids attached to it now. So for each of those, you'll times it by 2, and that's a total of 96 sister chromatids. Okay, so let's move on to part number 3. So this is asking what's happening at the very end of telophase. So you need to know what's going on at that point. So DNA replication has occurred. Maternal and paternal chromosomes have divided into individual cells. So at this point we have 2 separate cells, and the pairs have separated, okay? So the total number of chromosomes per cell. So there's 2 cells, and each of those are going to have half of the number of chromosomes that you started with, okay? So we started with 48, and we're going to divide that by 2. So each one of these, one will go to one cell and one will go to the other. That's the beauty of having 2 copies of each. Okay, and then homologous chromosome pairs. So we know here that the maternal and paternal homologous sets are separated. So you no longer have homologous chromosomes within the cell. Okay, so these have all been split right in half. One has gone to one side and one has gone to the other. Okay, so that's going to be 0. And then finally, how many sister chromatids are in each one? So we said from the first part of this, if we go back a few slides, part 3, at the end of telophase, We said each of these have 24 chromosomes within them, right? And each of those chromosomes, as we said, DNA replication has occurred, so each of those chromosomes have two sister chromatids. So you're going to multiply 24 by 2, and that will be 48. Okay, I'm going to let you figure out the last two parts of that question on your own. Um, in order to answer these, you need to know the difference between between homologous chromosomes and sister chromatids. So homologous chromosomes are similar, sister chromatids are identical, and you really need to know what's happening at each phase of meiosis. So meiosis comes after replication in the cell cycle, um, or the S phase, so you need to know what's happening in each one of those with the chromosome molecules themselves and the sister chromatids, if they're separating or not through the centromere. I hope this was helpful.